Yes. Neha, you have the introduction.
Yes.
Okay. So
Yes.
we can start in 2 minutes. Okay. You prepared. Be prepared because if Liberty come after a few minutes, so we can start.
Yeah. Okay.
Okay. Wait for one or 2 minutes because in 11 we will start. Tara ma'am, you have got the questions for the interview. Okay. It's a positive.
Yeah, they're very common things.
Thank you. Okay, Liberty, I just unmute you.
Hi. Share your views also. So if you have something specific during the interview.
Sorry?
Okay. No.
Oh.
Sorry.
Sorry,
Hi,
Neha?
Liberty. Ma'am, I'm requesting if you have something specific to tell to our viewers and to all the young scientists.
Okay.
Hello?
Before or after the talk?
Yes.
You want me to start it before the talk or after the talk?
After the talk.
Yeah,
After your talk, after the question
interview.
answer, then there's a different section of the interview, and Soma will take that interview. That time you can.
Okay.
Hello, Liberty. Liberty, are you ready?
Yes. I'm trying to find the
Tara, ma'am, it's a Dr. Satyavathi or Dr. Tara. What is the prefer for you?
Tara is fine.
Tara, I think Dr. Tara is well fine.
Yeah, that is easy to spell also.
I'm sorry. Okay, yeah, I have it.
Okay, so we officially start because it's 11 a.m. and we should start. Okay.
One second, I'm coming.
Okay, no problem.
From Bali.
Okay. Good morning, everyone, or good evening to others because it's a different time zone. Liberty and Jacinta is for her it is good night or good evening. I'm very happy to present today our speaker, Dr. Tara Satyavathi. Ma'am will working on millets, and Neha, Jacinta, Liberty, and Soma is a team of our Bioingene, and Liberty going to introduce our speaker. Neha and Jacinta will take care of the question answer round. So those are viewing in Zoom. Please ask the specific question to the talk related in that particular box. It is Q&A box. Non-unrelated questions like certificate and all, we are going to focus these questions after the talk. Those are the viewing it on from the YouTube. Please go and comment your question in the comment box so we can pick up the question for the ma'am after her talk. So Liberty, it's to you. Please go and introduce our speaker.
Sure. Thank you, everyone. Today we'll be hearing from Dr. Tara, who completed her Ph.D. in 1998 from the Indian Agricultural Research Institute in New Delhi. Presently, she is working as project coordinator at ICAR, AICRP on Pearl Millet. She was principal scientist in the Division of Genetics, IARI, New Delhi. Her research mainly focuses on quality parametrics in rice, crossability relationship and made hybridization in Brassica species, genetic improvement, development of minimal descriptor, national test guidelines, reference varieties for DUS testing in soybean, use of marker technologies like AFLP, SSRs in discerning diversity in soybean, identification of QTLs for seed coat permeability, breeding for bold seeded soybean with improved seed germination. She has reoriented Pearl Millet program at IARI, developed hybrids and varieties, identified QTLs for downy mildew resistance, agronomically important traits, developed and identified high grain iron and zinc Pearl Millet lines. She has genetically looked into genetic stocks for high lysine, high tryptophan, white grains, etc. She has developed a number of mapping populations for economically important traits like downy mildew, thermotolerance, along with a number of white-colored Pearl Millet lines that are of use in the food industry. As a project coordinator, she has introduced minimum iron and zinc content in the promotion criteria in varietal release policy for the first time ever in the world, culminating in release of 15 hybrids and varieties with minimum 42 ppm iron and 32 ppm zinc, leading to the nutritional security of rural women and children. Under her leadership, research efforts addressing recidity in Pearl Millet and development of Pearl Millet materials for arid zones were taken up. Dr. Tara received many awards, including the Outstanding Woman Scientist Award in 2016, congratulations, Outstanding Millet Scientist Award in 2018, Outstanding Research Contribution in Pearl Millet Improvement in the Country 2018, and the MS Woman Scientist Award in 2019, along with many best research papers and poster awards. She has published 88 research papers in reputed national and international journals. She has written 6 books and handled 20 research projects financed by DAC, the Government of India, DBT, AP CES Fund, GLDC Innovation Fund, BMGF, and ICAR. She has successfully guided 3 students for PhD degrees and 8 students for MSc degrees. Thank you so much, Dr. Tara, for being here today.
Ma'am, I'm requesting you to screen share your screen.
Sure. So the screen is visible?
Yes, it's fine. It's nice.
Okay. First of all, I thank the organizers for giving me this opportunity to share my views and also to make public what we have done related to biofortification in Pearl Millet. Just now you have heard from Liberty, which was mentioning about the things what we have done in for the first time in the world for Pearl Millet. So today I'm going to talk about the journey from biofortification in food crops to fixing benchmarks for micronutrient content in varietal identification and release of Pearl Millet. So if we see the present day challenges we see in agriculture or any field as such today, we are faced with the overbulging population, global warming, water scarcity, and malnutrition. And these are the challenges, common global challenges, and many people from different disciplines, not only in agriculture, medicine, and other fields, are also trying to address these global challenges. So today we are talking of biofortification, and this is related to nutrition. And if you talk about micronutrient malnutrition, this is one of the global challenges. It is nothing but also called as hidden hunger. It is due to the inadequate accessibility of minerals and vitamins in our diets. Micronutrients, though, as the term mentions, they are required in very less quantity. They play a crucial role in human health. In early 90s, this problem commanded global attention, and it was alarming, very alarming in developing countries who are predominantly based on cereal-based diets. And 2/3 of the population in the world depends on cereal and sugar crops-based food, which mostly lack essential micronutrients. Now, some of the facts which I want to put before you, there are 16 essential minerals which are essentially required for humans, of which 5 minerals, namely iodine, iron, zinc, calcium, and selenium, are limiting in our food, resulting in deficiency. And globally, 1 out of every 3 persons is suffering from ill health due to deficiency of one or the other micronutrients which I mentioned just now. Among all these micronutrients, iron and zinc deficiency accounts for more than 2 billion globally, and most susceptible being the pregnant women and children below the age of 5. And 1/3 of the 2 billion people suffering from micronutrient malnourishment around the world is from India alone. And in India, the annual losses due to iron deficiency is nearly 4 million disability adjusted life years. And for zinc, it is about 2.8 million disability adjusted life years. So this is the importance of these micronutrients. If you see, just now I mentioned, there are micronutrients are there which are essential. On a whole, if you see, we need 51 essential nutrients for sustaining the human life. The 3 most important things: air, water, oxygen, the energy, that is oxygen, water, carbohydrates, 9 amino acids: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. 2 fatty acids: linoleic and linolenic acids. 7 macro elements: sodium, potassium, calcium, magnesium, sulfur, phosphorus, and chlorine. 17 micro elements, of which I highlighted 2 here: iron and zinc, about which I am going to speak today, and 13 vitamins, including A, D, E, K, C, and vitamin D complex. And just now I mentioned that globally 1 in every 3 persons is malnourished, and we are in the middle of this global crisis. If you look into the consequences of this micronutrient malnutrition, they cause higher morbidity, higher mortality, lower cognitive ability, lower work productivity, impaired growth, and impaired reproduction. People may be wondering why she's talking about all these things, because my talk is something related to Pearl Millet biofortification. Yes, with this introduction thing, I just wanted to take you along why we are going for biofortification of the food crops, and from there how we have come to the setting up of benchmarks for release and identification and release of cultivars. And hidden hunger, it affects across the life in all the different stages, right from starting in the newborns to children, adolescents, pregnant women, adults, and seniors. The kind of effects it puts on different stages is: in newborns, it leads to low birth weight, high mortality, reduced mental development, and the child will be often ill. And in children, it leads to stunting, they fall often ill of the small stature, high mortality, and low resilience. And in adults, they'll be stunted with reduced mental and physical performance. And in pregnant ladies, it leads to high mortality, frequent perinatal complications, lower performance, and in adults, they'll be poor, malnourished and limited productivity. And seniors, they have lower life expectancy and they often fall chronically ill. So none of the life stage is without these hidden hunger effects. So one has to take care that in all through our life stages, we have to have proper nutrition to avoid all these ill effects. Now, nutrition as a first line of defense. We have already witnessed and are going through this corona pandemic. Now everyone realizes what is the importance of immunity and from where this immunity is coming. And we know that whatever we are eating, whatever we take, imparts to our health and immunity. So there is no doubt to say that nutrition is the first line of defense. And food systems are as much a part of a future solution as they are part of the current problem. So if we have any problem, it is also because of the food habits. And if we have any solution, it also is coming from the food habits. So everything lies in the food we take. And the most immediate frontline defense for people against infections is a healthy immunity system. And essential micronutrients such as zinc, vitamin A, and iron have a crucial role to play in building an immune system. And efforts to deliver micronutrients to vulnerable populations have used strategies such as biofortification, which use conventional plant breeding methods to increase the micronutrient content in the staple crops that are commonly produced and eaten every day. So biofortification is targeting those crops which are being consumed daily. And biofortification is shown to improve not only the micronutrient status, but also health outcomes, including cognitive and physical function, stronger immune systems, and lower morbidity to a range of diseases. And we have already seen in the world that food fortification is already in place. Worldwide, salt is being fortified with iodine. In countries like Zambia, sugar is fortified with vitamin A. In Bangladesh, vegetable oil is fortified with vitamin A. India, wheat flour is fortified with iron and folic acid. African countries with these things. And many other countries have different products which are being fortified with essential nutrients. Now, before I go into the thing, I just wanted to make a clear distinction between what is fortification and biofortification. Because this group is a wide, varied group comprising students, faculty, teachers, and many people. So for the benefit of the audience, I'm trying to bring out the difference between fortification and biofortification. Fortification is the practice of deliberately increasing the content of an essential micronutrient, that is vitamins and minerals, in a food so as to improve the nutritional quality of the food supply and provide a public health benefit with minimal risk to the health. Whereas biofortification is the process by which the nutritional quality of food crops is improved through agronomic practices, conventional plant breeding, or through modern biotechnology. Now, so biofortification aims to increase nutrient levels in crops during plant growth rather than through manual means during processing. So this is something which is being incorporated during the plant growth rather than the processing things. So it is a way, therefore, to reach populations where supplementation and conventional fortification activities may be difficult to implement or are limited. So if you look into the genesis of this word, it is coming from Greek word bios means life, and a Latin word fortificare means making strong. So This is a combination of a Greek and Latin word, and it is defined as the breeding of food crops that are rich in bioavailable micronutrients. And this concept was given by Howard Boyce when he had made a study when he was at IARI in Washington. And they have made a study and found out that the difference of health between rich and poor is based on the level of non-staple food consumption. Staple food means either rice, wheat, or maize, or something which covers major chunk of your energy in the form of carbohydrate. But the non-staple food means something like fruits, vegetables, dry fruits, and all these things which are rich in vitamins, minerals, etc. So his study concluded that mineral and vitamin deficiencies and not the calories were the main constraints to better nutrition and therefore to healthy and economically productive lives. So based on the report he generated from his study, it made people think, what if we could make plants to do some work for us? In other words, if we can produce new variety of seed that is higher in iron or zinc. In addition, farmers and consumers would not see or taste that extra iron or zinc, but their intakes would be substantially increased if it is combined with the staple food. And small farmers they grow the nutritious staple food and also consume it. And what if plants needed minerals for their nutrition, not only for human health, even these micronutrients are essential for the plant health also. And research has shown that seedlings with higher zinc content are more vigorous and viable, not only in wheat, similar kind of results are observed in different crops as well. And therefore, the intersection between nutrition and agriculture suggested that if we could find a way to increase the vitamin and mineral content of staple foods grown and consumed by smallholder farmers, we could solve the problem of hidden hunger. And thus, the intersection between nutrition and agriculture led to biofortification. So this is what about biofortification. Now, if you look into the science of biofortification, conventional plant breeding began hundreds of years ago, and every farmer chooses the best looking plants and seeds and he saves them for the next year's planting. And as the science of genetics was better understood, plant breeders were able to select certain desirable traits in a plant to create improved varieties of plants. Now, the projects of biofortification around the world focus on three crucial micronutrients that are limited in diets of the poor. There are three. There are 1, vitamin A, number 2, iron, and 3, zinc, and breed them into the key food crops. So when we talk of breeding for biofortification, the steps we have to follow would be first step in developing a biofortified crop is to screen the existing crop varieties and accessions in the global germplasm collection to evaluate the extent of genetic diversity and then identify your sources, maybe with the higher content or the lower content. The source of genetic variation is important for the next step in breeding because almost all the students of plant breeding very well know and understand the importance of genetic variation, creation of it, and then selection of the variants. So pre-breed, if it is available in some wild material, then pre-breeding is required if micronutrient variation is discovered in a strategic gene. And if micronutrient variation is present in the gene pool, materials may be used directly to develop new varieties. Then parent lines with high vitamin or mineral levels are cross several generations to produce plants with desired micronutrient content and other traits important for acceptance by farmers and consumers. Then once we have got that material with high desired micronutrient content, the next step is to thoroughly test it. So newly selected micronutrient dense lines in experimental station in farmers' fields. The plants are evaluated for their yield and other agronomic traits, including tolerance to major pests and diseases. This is what we also do in the normal development of any crop variety. These evaluations are done in comparison to the best available local cultivars. their influence of varying climatic and soil conditions is also studied, as well as performance and the different agronomic management practices is also studied. And this part is done through multi-locational evaluation. Then the seeds of the best performing materials are then multiplied and made available as public goods to national governments, which officially release the new crop varieties and are supplied to the farmers. So before we go for breeding for better nutrition, there are certain points which needs to be considered. The first point is aim at combining high nutrient density with high yields and high profitability. If you just blindly breed for high micronutrient content, there will be no takers. Even if you go and say to the farmer, see this particular variety has 1000 ppm of iron, he will not take it. Unless and until there is high yield, which in turn results in good economic returns, no farmer is going to take your variety. So the first and foremost step is to combine the high nutrient density with high yields for high profitability. Then the micronutrient enrichment levels. Yes, we started combining these two traits, but then you should select for micronutrient, enhanced micronutrient levels along with the yields. Then third, you have to look for the stability of the micronutrient enriched traits. Then efficacy must be demonstrated, and then only the so-called biofortified variety or crop will be adopted by the farmers and then by the consumers. So if you look into different breeding approaches for biofortification, you can either go by conventional approaches or through biotechnological approaches. So if you look into the process, first we go for screening of the genetic diversity for micronutrients in the available germplasm. We select that material. We go for allele discovery or through routine conventional plant breeding. We go for identifying of the improved cultivar with increased micronutrient content. Or by using different techniques like QTL analysis, you identify the trait-specific molecular markers, or you identify the gene, then allele discovery, use them through different marker techniques, and then go for development of the improved cultivar. Or you can do the genomic analysis, go for the gene discovery, take the help of bioinformatics and functional analysis, again come back to the allele discovery, maybe through the normal breeding or through the genetic engineering, you can come down to an improved cultivar. So once the challenge in biofortification is putting together all of the pieces of the puzzle to create a micronutrient-rich crop that is acceptable to both farmers and consumers. For farmers, it should be economically profitable along with high yield. For consumers, it should be acceptable in the original form without affecting their established taste and color and all other factors, organoleptic factors. So the advantages are, it is built on what poor households grow and eat, that is on staple foods. One-time investment to develop seeds that fortify themselves, keeping recurrent costs low, and the germplasm, that is the living tissue from which the plants can be grown, can be shared globally, making it highly cost-effective. Otherwise, if you have to synthesize artificially, you have to go for manufacturing or other things. Because it is a living tissue, it can replicate in terms of through breeding, it can be shared and it can be used. Biofortification is sustainable. Long after people stop thinking about biofortification, farmers continue to grow and eat their biofortified crops. Biofortification reaches the country's most vulnerable people living in remote rural areas with no access or money for commercially marketed fortified foods. And biofortification produces high yields in an environmentally friendly way, no side effect on the environment. And it is one solution to reduce hidden hunger worldwide. And it is the process by which the nutritional quality of food crops is improved through agronomic practices, conventional plant breeding, or modern biotechnological tools. So there are many biofortification projects taken up all over the world. Some of the things are here mentioning here. One is iron biofortification. It is used in rice, beans, sweet potato, millets, cassava, and legumes. Zinc biofortification done in case of wheat, rice, beans, sweet potato, and maize. Provitamin E or carotenoid biofortification used in sweet potato, maize, and cassava. Amino acid and protein biofortification in case of sorghum and cassava. In India, biofortification projects were funded by DBT and ICAR. At the world level, it was done by Harvard Plus. At national level, both by DBT and in ICAR, there is a special CRP on biofortification, which includes major crops like rice, wheat, maize, pearl millet, small millet, and sorghum. And today we have, there are biofortified varieties released in more than 30 countries and are being tested and grown in more than 40 countries. I'm just trying to go through some of the slides to show what are the different varieties released at different points of time across the world. I'm just going through this. In rice, biofortified, the micronutrient targeted was zinc. The country was Bangladesh. Brazil lead institutions were IARI, BRRI, Embrapa in 2013 and 14. Provitamin A, Philippines. You all know that golden rice developed at IARI. For iron, Bangladesh and India. Then iron, China. Many countries have started working in this direction on staple food crops. You all know about the golden rice. This is one of the solutions targeted for addressing vitamin A deficiency. For the sake of people who do not know about golden rice, it is a genetically engineered rice which can produce and accumulate beta carotene, that is provitamin A, inside the rice endosperm. So this is your normal white rice and this is your golden rice. I'm not going to explain these things, but for the sake of the students who would like to go through, they can go through the articles related to golden rice. Here, the genes were taken for phytoin synthase, lycopene synthase, and phytoin desaturases were taken from daffodils and Yersinia bacteria, and they were transferred to plasmids, and then transformed to agrobacterium, and then they got transgenic rice, which is rich in this beta carotene, and later it was termed as golden rice. There are different versions of golden rice available. First one was a prototype. It was having 1.6 micrograms per gram of golden rice, beta carotene. Then golden rice 1, after changing the promoter, they got 5 to 7 micrograms of beta carotene per gram. Then golden rice 2, it was the gene was changed from the daffodil to maize gene phytoin synthase, where they could get 31 micrograms of beta carotene per gram of the. So you can see the variants. This is the normal rice, golden rice 1 and golden rice 2. With increase in the content of the beta carotene, you can see the change in color also. That was at the national level. In India, micronutrient content efforts were made in different crops to increase these micronutrient contents. They also have fixed some benchmarks to develop rice varieties. For zinc, they have kept 28 ppm. For iron, they have kept 10 ppm. For protein, they have kept more than 10%. And we have different rice varieties like CR Dan at 310, which has 10.3% of the protein. This comes under a biofortified rice variety. Then zinc, 22.6 ppm. It is DRR Dan 45. For wheat, in the international level, these are the efforts taken by different countries for zinc. It was efforts were being made by India, Pakistan, China, Brazil, and other countries. And many biofortified wheat varieties with different levels of zinc were developed. And in ICR at the national level, we have WB02, which has zinc 42 ppm and iron 40 ppm. And then HPBW01, where iron is 40 ppm and zinc is 40.6 ppm, released by different centers under ICAR. In maize, the nutrient targeted was provitamin A. Different countries like Zambia, Nigeria, Brazil. Also in India, efforts were made by different institutions. The material developed in India for maize is Pusa Vivek Cure PM9 improved, where the provitamin A content is about 8.15 ppm, lysine is 2.67 ppm, tryptophan is 0.74%. And in Pusa HM4 improved, tryptophan is 0.91 and lysine is 3.62. Then Pusa HM8 improved, tryptophan is 1.06%, lysine is 4.18%. Pusa HM9 improved, it is tryptophan 0.68%, lysine 2.97%. Then at the international level in the crop sweet potato, provitamin A was targeted again. And you can see the orange sweet potato, which is rich in provitamin A. You can see the difference in the flesh color. The normal flesh of the sweet potato is yellowish in color, whereas if it is rich in provitamin A, you can see orange to dark orange color. That indicates the richness in provitamin A content. And you can see the kind of bread prepared from this sweet potato. In India, sweet potato Bhu Krishna was developed by CTCRA, where the anthocyanin pigment is about 90 mg per 100 grams. And then potato developed by CTCRA again. Here the beta carotene content is 14 mg per 100 grams. In cassava, then targeted nutrients were provitamin E and iron content. Targeted countries, these are the different countries which worked on these crops for these micronutrients. And for beans, targeted nutrient was iron, and it was worked out in African countries. And different crops for different micronutrients: lentil, iron, zinc; potato, banana, provitamin A; coffee, iron and zinc. Different countries started working on different micronutrients depending on the crop they consume majorly. And in India, not only developing micronutrient-rich content varieties, but also reducing anti-nutritional factors. That is also a kind of biofortification. In case of mustard, varieties like Pusa 00 mustard 31, varieties like means they have developed a variety which is having less than 2% of erucic acid and less than 30 ppm glucosinolates. And in Pusa mustard 30, the erucic acid is again less than 2%. In lentil, Pusa Ageti Masoor, here the iron content is 65 ppm. And in vegetables like cauliflower, Pusa Beta Kesari 1, beta carotene content is 8 to 10 ppm. And in fruits like pomegranate, Sholapur Lal, here iron content is 5.6 to 6.1 milligrams per 100 grams. Zinc is 0.64 to 0.69 mg per 100 grams. Vitamin C is 19.4 to 19 mg per 100 grams. Such is the extent of variability in the micronutrient content and richness of these nutrients in the pomegranate here. So this is one of the slides which I took from Harvest Plus site. This shows that the different biofortified crossbred all over the country, all over the world, are able to address and meet daily requirement of these micronutrients. For example, iron Pearl Millet, it is able to provide up to 80% of the daily iron needs. And the benefits are they are high yielding and disease resistance and drought tolerant. Similarly, for different crops, different characters were mentioned along with the extent of the nutritional benefit they are going to provide. Now, since my talk is on the related to benchmark level, now I'm going to talk about these different benchmarks and in different crops and then come down to Pearl Millet. I have shown you some varieties which have been released, developed and released by different institutes in the country and also in the world. Now, this is one slide which I'm going to speak about the status of the baselines and levels achieved in different crops in India. For rice, for zinc, the baseline which was about 12 to 16 ppm and the levels achieved were 20 ppm. For protein, 78%, more than 10% was achieved. For wheat, the level fixed was 28 to 32 ppm for iron, but the level achieved is more than 38 ppm. For zinc is 30 to 32 ppm, achievement is more than 40. For maize, lysine targeted is 1.5 to 2, the achievement is more than 2.5%. For pearl millet, 30 to 35, more than 40 ppm for zinc and more than 70 ppm for iron. Similarly, for vegetable crops like cauliflower, beta carotene, it was very negligible. They could come up to 8 ppm, more than 8 ppm. Sweet potato, I just now showed, anthocyanin content, they have achieved to more than 80 mg per 100 grams of the pulp. A pomegranate, vitamin C, this much iron and zinc. And I also said that reduction in the anti-nutritional factors is also a kind of thing which they're looking for. In mustard, they could obtain glucosinolates reducing from 120 ppm to less than 30 ppm and erucic acid reducing from 40% to less than 2%. And in soybean Punnet's inhibitor reducing from 30 to 45 mg to almost negligible. So this is all about the kind of different kinds of biofortification taken up in different crops. Now I'm coming to the topic of millets or pearl millet. Before I go directly into pearl millet, many people confuse millets. They think that sorghum, pearl millet, millets are either combinedly they call sorghum, pearl millet, small millets, everything as millet. But there are different kinds of millets. Millets is a common term used for representing this group of grains. Millets are one of the earliest grains that have been cultivated and consumed by the people in India and world. We have different kinds of millets, major millets and minor millets. Major millets, they include pearl millet, sorghum and finger millet. Minor millets, they include foxtail millet, proso millet, kodo millet, barnyard millet and little millet. Pearl millet and sorghum are the important food grain crops in India after rice, wheat and maize in terms of area and production. And finger millet is also one of the major millet which is grown and is consumed in major parts in states like Karnataka, Telangana and Uttarakhand. And nowadays everyone is talking of millets. Whenever people talk of immunity and health, people first come out with millets. And if you just want to know what are these millets and how they are beneficial and why the millet production is beneficial, the millets are beneficial because they provide nutritious food, feed and fodder. They are less water consuming than other cereals. They grow faster, putting less stress on the environment, less incidence of insects, pests and diseases. They can grow even on very poor and marginal lands. And they provide complete food and nutritional security. They will supplement the existing income of the farmers by reducing groundwater extraction. They combat desertification, increase farmer resilience to drought brought on by climate change. And United Nations Food and Agriculture Organization has declared year 2023 as International Year of Millets. And year 2018 was declared as National Year of Millets in India. And millets have been renamed as nutri-cereals by Government of India through a gazette notification issued on 13 April 2018. And these nutri-cereals include sorghum, bajra, finger millet, and all these millets. And if you look into the health benefits of these nutri-cereals, they are beneficial in detoxifying the body. They lower the bad cholesterol level. They prevent the onset of breast cancer. They help to prevent type 2 diabetes. They're effective in reducing blood pressure. They help to protect against heart disease, aid in treating respiratory conditions such as asthma. They help to optimize kidney, liver, and immune system. They reduce risk of gastrointestinal conditions like gastric ulcers and colon cancers. And they eliminate problems like constipation, excess gas, bloating, and cramping. Now, if you look into the condition and the statistics of millets, millet consumption in India is trending improvement for better health through functional. Now people have started understanding the importance of the nutritional value of these millets. The consumption pattern started including millets in their diets, and therefore the consumption is increasing continuously in India. And if you look into the trends in the health and wellness industry, the tendency of people has changed. That is, they are moving from reactive to proactive. That is, from better to avoid treatment than going for treatment. It is better to go for prevention. So how they are going to prevent? By taking healthy diet. And how they are coming to get this healthy diet? By diversifying their diets. So going from just their traditional, the much more recently habituated condition of eating rice and wheat, people have started moving to taking up even millets, and they are trying to diversify their diets. And there is a big opportunity in the processed food sector. If you look into this, most of the processed food is only this much, semi-processed is this much, and this much is remaining for the unprocessed food sector. And almost all the processed food products are either coming from wheat, maize, or rice. And this much of unprocessed food segment is there, which can be explored by millets. And I said millets need very less water. If you look into the water requirement, sugarcane takes up almost 2,100 kgs of water to produce 1 kg of sugarcane. Rice takes 1,250 liters of water to produce 1 kg of rice. Whereas pearl millet or other millets, they take 350 to 400 kg of water or 1 kg of grain. You can see the extent of difference, how much of water is saved by growing these crops. And nowadays, grain drain is happening. Till now, we have been hearing about brain drain in India. Now, grain drain is happening because the cheapest cereals in India are the most expensive ones in US. Most of the people in US might be knowing about quinoa, millets. These are very premiumly priced products. I think for 1 pound, you people are selling about $5 to $8. Whereas the same pearl millet, other things are available for a minimum of 20 rupees per kg. Compared with the $10, that is around 63 to 630 to 700 rupees per kg in US, whereas it is only 20 rupees per kg in India. And most of these millets are being now exported or taken by foreign countries, and they are taken up at a premium price in these markets. Now, when I started my talk, I said that climate change is also one of the biggest global threats. I said micronutrient malnutrition and climate change. If you look into the different crops, this is the thing which is showing the temperature. Wheat can withstand a temperature of 30 degrees. Rice can withstand a temperature of 32. Maize maximum up to 40. Pearl millet is one which can withstand higher temperatures. It can even flower and set seed at as high temperatures as 42 degrees centigrade. And if you see the conditions where these different crops grow, rice needs nearly 900 to 2500 millimeters annual rainfall, maize 500 to 800 annual rainfall, wheat 450 to 659, whereas pearl millet can grow even from areas receiving 200 millimeters to 600 millimeters rainfall. And this is a nutri-rich crop, nutri-cereal. Coming to India and pearl millet, it is grown in an area of 6.93 million hectares with a production of 8.61 million tons and a productivity of 1243 kgs per hectare. The major pearl millet growing states are Rajasthan, Maharashtra, Gujarat, UP, MP, Haryana, Karnataka, all these states. Like in any other crop, pearl millet is also having different agroclimatic conditions suitable in different zones of the country. We have three different agroclimatic conditions, zones for pearl millet. One is A1, other is A and B. Majority of the area is in A1 zone, which is represented by western part of the country, comprising those areas which receive less than 400 annual rainfall, covering western Rajasthan, Gujarat, and parts, very little part of ISAR. And there is another area called as another agroclimatic zone called as A, which comprises those states and areas receiving more than 400 to 1000 annual rainfall. And here the crop grows in a very good condition. And another agroclimatic zone represented here is the southern part of the country, which is B zone. And here the area is about 1.14 million hectares. And the climatic conditions and the requirements of the varieties and hybrids for these different zones are different. If you look into the area production and productivity, this is a line which shows a constant continuous decline in area. And this is the line which is showing the productivity. In spite of the constant decline in area, you can see a constant and continuous increase in the productivity of the crop. So even though the area has reduced, the production and productivity are still higher because there is a continuous increase in the productivity. And this was attained because of the high yielding hybrids which were developed in pearl millet. Just now I mentioned that we have three different zones, A1 zone, A zone and B zone. These are the different areas and different material cultivars suitable for these areas. There also we have one more segment, that is summer segment, where it is nearly 0.4 million hectares. This is grown in Gujarat and those areas immediately after the harvest of potato. The soil will be rich in nutrients and it is grown up in summer months because the temperature is high. Nutrients are available in the soil and it is grown in those areas where they have irrigation facility. So the crops, the pearl millet crop comes in a beautiful way, very good high yields. In normal condition, we get about 3.5 to 4 tons per hectare. Whereas in this summer pearl millet, the yields are as high as about 6 to 6.5 million tons per hectare. That is the extent of yield we get in summer pearl millet. Just now I mentioned that pearl millet is also mentioned as a nutri-cereal. It is rich in energy, carbohydrate, protein, fat and all these things. It is a gluten-free grain with low proline fraction and it is ideal for people with gluten allergy. And Government of India has made efforts to include pearl millet and other millets in the public distribution system. And year 2018 is named as Year of Millets. This is the nutritional composition of pearl millet along with other millets compared to rice and wheat. Without any doubt, we can say that it is nutritionally superior to other grains we have. These are the different major nutrients, carbohydrates, proteins, and fats. Proteins are gluten-free, low in lysine. Because they are gluten-free, they are suitable for people with allergy, celiac disease, and this is the only food grain which retains its alkaline property even after it enters our gut, after enters the stomach, our digestive system. Remaining all other foods turn acidic. So if someone is having any problem, acidity or other gas problems, this is the best food which can be given to those people. It has low glycemic index. It is only 55. It is ideal for people suffering with diabetes. And it is having highest amount of fat. But if you see the composition of this fat, it has omega-6, that is linoleic acid, omega-9 and omega-3 fatty acids. Highest content of these. People run after puha. This is the rich source of all these things. It has a beautiful combination of different vitamins. See, in comparison with wheat, rice, sorghum, and maize, it has a good, beautiful composition of vitamins. And just to, because we're talking of biofortification, iron and zinc, I just wanted to show the audience that this is the endosperm or the germ, and this is the endosperm. Iron is present in this layer, alderon layer. Here, this is the iron and zinc is present here. Now, with this introduction of pearl millet, before coming to pearl millet, I spoke about different crops, the use of biofortification, why we should go for biofortification and all these things. Now coming to pearl millet and biofortification, why this topic of point of biofortification has come to pearl millet. If you look into the intake of pearl millet in growing regions where pearl millet is grown, 35% of the total intake of the calories, protein, iron, and zinc is coming from pearl millet. These are the main pearl millet growing states, and it has been studies have been made in these areas, and it has been seen that nearly 35% of the total intake is coming from pearl millet. Now, as I mentioned earlier, biofortification aims to address nutrient deficiencies as an integral part of the core breeding program, but there is a need to understand the potential impact of higher macronutrients on other important things. Now, enhanced nutrient content in new cultivars have to be achieved without any trade-off with higher productivity. That's what I said. Whenever we go for breeding for biofortification, the farmer should also be happy and consumer also should be happy. For instance, selection for increased micronutrient content may be expected to negatively affect yield or other important agronomic traits. But this will happen only if the genes that increase micronutrient content are linked with genes having a deleterious effect on the yield. So the breeder, when he or she thinks of breeding a variety with high iron or zinc, they should see that there are no negative linkages of these nutrient contents with yield. So this is what I have shown in the beginning. The same slide again I'm showing here. So as shown here, first we have to see the genetic diversity for the micronutrient content. In pearl millet, they have seen a large amount of variation existing in different kinds of material. So this is the extent of information. Still some more references have been added, but I'm just trying to show here. This is the range for iron starting from 29.4 ppm to 87.9. In some cases it is 102, as low as 18 to as high as 139 ppm. Similarly for zinc also you can see as low as 13.5 to as high as 89.89. The large amount of variability is present in different kinds of material available in pearl millet. And it was also observed that the kind of association of iron with the different parameters. With zinc, it was shown to be significant and positive. It has been shown to have a slight negative or non-significant association with yield, then zinc with iron, zinc with grain yield, and many other parameters have been shown. And it is also shown in different studies that there is a direct positive correlation between iron and zinc. Higher the iron content, higher the zinc content also was observed in some studies. So with this background information, people started thinking of breeding for higher micronutrient content for iron and zinc in Pearl Millet. Now, as a breeder, when I think of breeding for iron and zinc content, okay, I'll make process, but how do I know how much iron or zinc is there in the material which I am breeding for? That means I need to go for phenotyping of that trait. But this we have, that means it has to be screened. So a large amount of germplasm was screened, and for this a XRF tool was available, which is time saving and effective way of measuring the iron and zinc content. This is a XRF. It uses non-destructive sampling. The cost is about only $2 per sample. It is restricted for only for iron and zinc for the time being. And the amount of data which is obtained for, through XRF data when compared with ICP, there is a correlation to an extent of 0.69 to 0.97, and for zinc it is 0.60 to 0.98. This is safe and simple to operate. It handles more number of samples per day, and it is relatively cheaper compared to the existing tools. Now, I said that at the international level, Harvest Plus has taken a lead in developing micronutrient-rich crops or biofortified crops. Same Harvest Plus has also taken a lead in Pearl Millet in developing iron-rich or zinc-rich varieties in Pearl Millet in the initial stages. And the first wave of efforts by Harvest Plus led to the development of the open pollinated variety called Untitled, which was released in India in 2012. And then a second wave of release came from Harvest Plus in terms of a hybrid, which was released in 2014, that is MH1201. And another hybrid was also released, which was released in 2016. This is from the part of Harvest Plus. And this is the variety, the first variety, Dhanshakti, which was released by collaborative efforts of ICAR with MPKV Dule. And this was tested under ICAR, AICRP and Pearl Millet, and also released, it was released through AICRP and Pearl Millet at the national level. Now, coming to the efforts of ICAR, I said that there is a separate consortia research platform on biofortification led by ICAR, in which Pearl Millet is also one of the crops, targeted crops. And in Pearl Millet, these are the two objectives: to develop Pearl Millet genotypes for high zinc and iron through conventional breeding, and identify genomic regions associated for enhanced micronutrients through mapping and association mapping, and also develop value-added products. So there are different centers, around 6 centers are there under this project. And under this project, along with the material obtained through Harvest Plus program by the different centers, the following 1 hybrid and 1 variety and 6 hybrids were released in different points of time in Pearl Millet. And here the varietal release policy is like in any other crop. In Pearl Millet also we have 3-type system of testing. The first year will be initial varietal trial or initial hybrid trial. This next year will be advanced hybrid trial or advanced varietal trial 1, and then third year will be advanced varietal trial 2 or advanced hybrid trial 2. Now, for development and release of varieties, all the material has to be passed through AICRP in Pearl Millet. And in Pearl Millet, After the biofortification plant breeding and Pearl Millet was started, a separate trial of high iron and zinc was started in Pearl Millet. And this is how these 6 hybrids were released from that trial. If you look into the iron and zinc content here, it is 73 ppm iron, zinc is 41 ppm in HHB 299. 77 ppm iron, 39 ppm zinc. Similarly, higher amounts of iron and higher amounts of zinc are seen here. But if you see the yield levels, the highest grain yield they could get is, means the average in the experiments they could get is only 3.2 tons. It is 3.1 ton or 3.2 tons. But if you see in the normal coordinated trials, the yields are somewhere around 3.5 to 4 tons per hectare. These high iron and zinc varieties, though they are having high iron and zinc, they are not matching with the regular hybrids and varieties in terms of yield. When I started speaking about biofortification, I said that the farmer should also be happy and consumer also should be happy. Now, in spite of having high iron and zinc, if they are not able to give high yield, these yields are not very low, but they are not very high as. So, if we have to combine high yield and high micronutrient content, then we should have a different strategy. Now, also, when these hybrids and varieties are released as biofortified varieties, in the market, the farmer is not going to get any high premium price because there is no segregation in the market level. Number one. Number two, even in the market, you have any system, there is no system to screen that this is a high iron variety, this is a low iron variety. Because there is no premium price available, there is no system of segregating high iron material with the low iron material. And number three, the consumer is also not able to differentiate which one is high iron material, which one is low iron material. And in the long run, we will be coming up only with varieties, but without market, without translation to the without being taken to the end user or the consumer. So we thought that because we have large amount of variability available with us, and there are established procedures of developing high iron pearl millet or high zinc pearl millet, and also it has been demonstrated that we can develop the materials with yield and these increased nutrients. So we thought, why not we club these high iron, these micronutrients into our main breeding program. That is, we started mainstreaming this micronutrient content into our pearl millet breeding programs. These are the high iron hybrids which I just mentioned in the previous slide. This is AHB 1200. This is HHB 299. This is field view. Then HHB 311. This is coming from ICAR. AHB 1269 coming from Aurangabad. RHB 234 coming from Durgapura, Jaipur. Again, RHB 233 coming from Durgapura, Jaipur. Now, those people, I'll just come back to these slides. Yeah. Whenever I have shown you biofortified hybrids and varieties. Now, when we give this material to the farmers to grow, the farmers have some questions. They say, you are giving us biofortified material to grow. Whether this iron and zinc content of any cultivar will be the same, regardless of the environment where they are grown. That means they are asking us whether there will be differences in these content. Then whether iron and zinc content of any cultivar depends on the inherent soil iron and zinc content. Whether grains produced in environments higher grain yield will have low iron and zinc content compared to those produced in environments with lesser grain yield. Whether the bioavailability of iron and zinc in the grains from cultivars with high levels of these nutrients will be more from those with the lower levels of nutrients. Yes, these are not only questions put for the farmers, some breeders and consumers also, the policymakers also have put. Yes, whether there is iron and zinc content difference depending on the environment, the iron and zinc content vary. That is why when we thought of including these traits into the main breeding program, we thought they should be tested in multi-locations. That is why we have made them a part of our testing criteria. And also, the other question they had put is whether iron and zinc content of any cultivar depends on the inherent soil iron zinc content. It does not depend on it does not totally depend on this iron and zinc content unless and until the soils are deficient in those nutrients. If it is a normal soil, it will not be affected. But if the soils are hungry, then definitely the iron and zinc content will be affected. Whether grain produced in environment with higher grain yield will be varied for the low grain yield, lesser grain yield environment. There is no difference between the higher grain yield environment and a lower grain yield environment in the iron and zinc content. And also there was a question whether bioavailability of iron and zinc will be higher in those coming with higher micronutrients compared to those of the lesser micronutrients. Bioavailability does not depend on the quantity of the nutrient available in the variety. It is not affected by that. So with all these things in the mind, this point we have started mainstreaming micronutrient content in our Pearl Millet breeding program. For this, the base study was taken by AICRP and Pearl Millet and ICAR in 2016, where a multi-location trial of 18 open pollinated varieties and 122 hybrids was conducted in 2 years over different locations, and the iron content ranged from 42 to 67 mg per kg in varieties and 31 to 61 in hybrids, and zinc content varied from 37 to 52 and 32 to 52 in hybrids. And these 122 hybrids and 18 open pollinated varieties are nothing but the varieties and hybrids which were released through the system over a number of years. So in one way, the entire amount of variability present in the released varieties and hybrids were assessed over years and over decades. So this gave us an idea of the amount of micronutrients, both iron and zinc, available in the released material. So based on this, the benchmark levels were derived, and then a landmark decision was taken in 2017 in our annual group meeting, where we fixed that iron content, a minimum of 42 ppm of iron content should be there, and minimum of 32 ppm zinc content should be there for promotion in All India coordinated trials and for identification and release of the material. I mentioned that we have a 3-tier system of testing. So in any crop, whether it is rice or wheat or maize or pearl millet or chickpea or any other crop, we have these 3 years of testing. So for promoting any entry from one year to the other, it will be tested across some standards with the checks and with the other material. And it will be promoted to next year only when it has superiority in yield and it shows resistance to existing diseases and insects. Till date, only crop flowering, diseases, insects, all these things are taken care. But nowhere a micronutrient, a nutritional quality trait was incorporated as one of the promotion criteria for promotion. The slides which I have shown previously where biofortified varieties were released in rice, wheat and other things, there it is for release under the category of biofortified varieties. But here I am speaking of the normal variety. So instead of categorizing a special group as biofortified or normal thing, we started incorporating this nutritional quality trait also along with the yield traits. So now if a Pearl Millet breeder has to develop a variety or a hybrid, he or she should keep in mind that along with yield and other desirable traits, these two traits also should be there, and minimum of 32 ppm zinc and 42 ppm iron should be available. And after that, we have materials released, identified for release. These are the four. The ones in black color are the normal hybrids. They released after our baseline was this benchmark was constituted. You can see iron is more than 42 ppm here, and zinc is more than 32 ppm here. Here, the ones which is showed in red color are the biofortified material because they are coming in a different trial. These are the normal hybrids without any biofortification tag, but now having more than the benchmark level. And in 2019, these are the hybrids released, where again you can see iron more than 42 ppm, zinc 32 ppm. Now this is the, till here I have shown about the released material, identified and released material. These slides which I am showing is the extent of variability we see in the trials, different materials present in the trials. You can see as low as 27 to as high as 100 ppm. This is in 2017, this is in 2018. For zone A, these are the different trials. This is the kind of variability you can find. For zone B, these are the different trials, and this is the extent of variability. This is the range. This is the range here. This is for iron content. Coming for the zinc content, you can see the extent of variability. This is the range here in 2017, 2018. For zone A, again, this is the range. This is the range in 2018. For zone B, 2017, this is the range, and this is in 2018. So now we have sufficient amount of variability for iron and zinc content across the trials and across the material. Recently, someone has put a question to us. Harvest Plus has put the level as 77 ppm. And why you people have taken only 42 ppm of iron? Why this much of difference is there? Just now I have shown the slides in iron content. You see, this is the extent of variability. And if you see the average, this is the thing, around 48, around 55, 42, 55. Here, here also it is the same. Unless and until we have higher amounts of micronutrient content present in the developed material or the material which is in the pipeline, we cannot increase the level to 70 or 77. And no variety will be identified for this. First of all, incorporating a nutritional trait as one of the promotion criteria itself is a big change. Again, to change it again for increased quantity, unless and until we build up the material with enhanced micronutrient content, we cannot go for a higher amount of the nutrients. Now, if we are having a biofortified tag, you have to have a special seed production program, and then it has to be continuously monitored that it has that much iron content or zinc content, and then the seed has to be specially produced and specially marketed. Now, by making this iron and zinc content as one of the mainstream breeding character, it is taken as a normal trait, just like any hybrid or variety is produced. The similar procedure is adapted for developing, producing the seed of those material, and then it is being sold. So in Pearl Millet, because it is a highly cross-pollinated crop, we have major role of hybrids. And in this, private sector has a very good role in developing the hybrids and marketing the seed. Though public sector hybrids are available, public sector lags compared to private sector in the aspect that public sector maintenance breeding is poor, whereas in private sector, seed production is taken on a very aggressive mode and is also marketed in a very aggressive mode, which is lacking in public sector. And now, once we have these hybrids and varieties with enhanced iron and zinc, how can we transfer them? That means the spread of these material, it can be taken on a large farm field trials like frontline demonstrations either through agriculture universities or KVKs, or then large scale production and procurement of these varieties and including them in the Anganwadi programs or including them in ICDS schemes or special government sponsored programs like NFSM and integrated child development program. They are providing window for public distribution system using these enhanced iron and zinc varieties and hybrids. And also develop technologies to increase shelf life of pearl millet flour and creating demand for increasing consumption of iron pearl millet and establishing market linkages for value added chain products. All these things can be taken up. And this is one effort where pearl millet, the nutritional properties of pearl millet are brought out in this booklet portion where it is nothing but pearl millet oriented, staple, healthy, all-inclusive nutrition. This is a publication brought out by Division of Biochemistry, IARI, highlighting the nutritional properties of pearl millet. And I mentioned just now that pearl millet is the first crop to incorporate iron and zinc content as one of the promotion criteria in our All India Coordinated Pearl Millet trials. And this is a certificate given by Harvest Plus, where they are mentioning that for contributing to the National Biofortification Partnership and also for setting iron pearl millet and establishing first global minimum standard for micronutrient in the cultivar release policy that has a significant and lasting positive impact on the mission of harvesters for improving human nutrition. So this is the first of its kind of step taken by the Pearl Millet group, and many of the crop coordinators have asked me how you have taken this decision to incorporate these nutritional traits into the promotion criteria. Just now I have told that we have screened the entire material released for both varieties and hybrids across locations and across years. And after seeing the data, after seeing the range of these nutrient contents in this material, then only we have come to a decision that this should be the minimum level of quantity for iron and this should be the minimum level for zinc. For iron, we have set as 42 ppm. For zinc, we have set as 32 ppm. For all his background work, most of the work was contributed from ICAR, where Dr. K.N. Rai and his group, supported by Harvest Plus, they have done wonderful piece of work in generating the data, and based on that, this decision could be taken. And now Pearl Millet stands to be the first crop to include nutritional traits in breeding, developing the hybrids and varieties which are rich in iron and zinc content. And this is how we are trying to contribute to nutritional security of the country. And coronavirus pandemic has highlighted the importance of good nutrition. And this is a critical wake-up call to rethink, redesign, and reprioritize our food systems to deliver nutritious and affordable food for all. That is how our pearl millet would fit into this, because it is nutritious, affordable to all, and it can be easily taken up. And policymakers, researchers, and agriculture sector need to come together to systematically solve the food divide that the pandemic has highlighted. So this is one of our baby steps towards ensuring good nutrition for all and providing immunity to everyone. Thank you for your attention.
Thank you, ma'am. Now, there is a question and answer deposit box. There is a, I think there's a lot of questions. You can choose randomly few, and later we go to the interview section.
Rajan Chaudhary has asked me a question that nutritional deficiencies can be addressed through biofortification of staple foods, which people eat most. And what is your opinion on biofortification of millets, which is not a major part of the common people's diet? So I think you might have got your answer in one of my slides where I showed that people who are dependent on pearl millet, nearly 35 to 40% of their calories, energy is coming from pearl millet. And it is being consumed in not only in India, it is one of the major staple food crop in Africa also where it is grown. Because most of the people in India are presently they eat only rice and wheat. But if you look into our traditional eating habits, we traditionally we eat different kinds of foods in different times of year. But now it has become a fashion that we should take only rice or wheat roti or other things. If you ask your grandparents or even your mother, they will tell you that they used to take different kinds of grains in different parts of the time because they are supposed to give us different kinds of minerals which are essential to build immunity to the changing conditions in different parts of the period. And now people have started realizing the importance of this millet and they're eating. If someone is not able to understand the importance of that, we cannot help them. But people are changing and they are taking and they are embracing millets in their diets. If you go to some 5-star restaurants in Gujarat, pearl millet is a delicacy in some restaurants, in 5-star restaurants, which is premiumly priced. For normal people, they may not be knowing the importance of this, but people who know the value, they are consuming it. And that is why this pearl millet biofortification has come into. I think I have shown 2-3 slides on that. You can get back to that. Sanjeev Kumar has asked me a question. Is the process of biofortification creates an extra burden on plant? And whether it will disturb the natural balance given by nature? Can you explain? It is not going to create any extra burden on the plant and it is not going to create any imbalance provided by the nature. Because if you remember, when I started the science of biofortification, I said that first we have to screen for the amount of existing variability. There is a large amount of variability available. That means already these things are available in the nature. We have started lately looking into these characters. Otherwise, they were available in the nature. So we are not creating any extra burden on this. That's why I said when we are breeding for high iron pearl millet or high zinc pearl millet, I said that we are trying to combine these traits along yield, not at the cost of yield. So these are well gelling together and they're going together. No burden on the plant or not they're creating any imbalance on nature. Someone, Dr. Rathod, asked me, any pearl millet ICAR biofortified hybrid released for cultivation in Maharashtra? Yes, there are 2 biofortified hybrids available in Maharashtra, AHB 1200 and AHB 1269. Any interested people can approach NARP Aurangabad. Dr. S.B. Pawar is the in charge of that, head of that unit. You can approach him. They can provide you the seed for that biofortified hybrid. Someone asked me, Fayaz Ahmad Dar, do we still need biofortification of millets and pseudo cereals? Although these crops are naturally enriched with essential micro and macronutrients. That is why we have mainstreamed these characters. We are not trying to breed biofortified varieties no more. We are trying to make them mainstream characters. So we will not have a separate category, but all the varieties and material coming from these 3D programs will naturally have high iron and zinc. Someone asked me about what is the current status of genome editing technology in millets for biofortification or stress tolerance. Right now, for biofortification, no one is going to use genome editing technology in Pearl Millet. But yes, genome editing technology is being used for uh, addressing the issues of rancidity in Pearl Millet, not for stress tolerance or for biofortification. Someone asked me whether there is any linkage drag found in Pearl Millet. We did not find any linkage drag. In some cases, there were some reports where there is high zinc content linked with low yield, but they are very few. And the answer to that is because you are able to find such segregants. If we have directed selection for increased nutrient content along with yield, that can be addressed. Someone anonymous attendee. At the time of polishing and milling, we remove most of the storage layers. Then what is the benefit of biofortifying if these layers are removed? The pearl millet and these millets are not going to be provided as polished grains. If you see the way these things are consumed, they are directly grinded, the powder is taken, and then it is consumed in used in preparations. So I don't think you are going to lose any of these nutrients. Someone asked me, I think Dr. Kiran Kumar, is there any possibility in field crops to enhance the micronutrient content in the grain through agronomic practices like repeated foliar spray of micronutrients at flowering stage or prior to flowering stage? There are reports that foliar spray, 4% ferrous sulfate, foliar spray, it is going to increase the yield, but we do not have at the reports which show that the micronutrient content is increased by spraying of these foliar sprays. Research has to be done to give answer to this. HHB 299 and HHB 311 are biofortified hybrids for HISAR. No, these are the hybrids coming from HISAR. These are the hybrids developed by HISAR group in collaboration with ICAR, Harvest Plus and AICRP and Pearl Millet. What is the amount of micro and macronutrients that are used in hybrid Pearl Millet? This is not the right kind of the question.
Ma'am, you take only one questions, just the last one question.
Any iron and zinc linked markers validated in Pearl Millet? Yes, there are reports, and one of my students at IARI has done association mapping. She has already given iron and zinc linked markers. One more student has done QTL mapping. That paper is in line, and also we have some publications along with the ICAR where QTL mapping for iron and zinc has been done, and already markers are available.
Okay. Yes, ma'am. Thank you. I also got many questions, and you have most of the questions you have covered in your slide, and now it is our request to you to join the short interview session. And are you ready, ma'am? Okay,
Sure.
so I hand it over Shoma. Shoma will conduct a short interview. Shoma.
Hello, ma'am. It was a very informative, very nice
Be a
presentation.
bit louder, ma'am.
Hello.
Yeah.
Okay. Ma'am, it was a very good, very nice, very informative presentation. Learned so much about biofortification and how we can increase the nutrients by so much in the crops. And I hope that pearl millet comes into the mainstream soon. More people accept it as a superfood. And congratulations on the work, on your work, on your to your team for the beautiful work they have done. Ma'am, we want to know more about you. Could you tell us about your journey in becoming a plant scientist?
Okay. Uh, I come from a middle class family, and as any of my contemporaries, I also aimed for becoming a human doctor. We used to have a common entrance for coming into biological stream and then getting selected for the medicine.
Mm.
Not clear for that. From right from beginning, I used to have that I should contribute something to the society. So I thought medicine was the best profession. Medical field.
Okay.
Coming into agriculture it was purely on the behest of my father. When I could not clear this medical entrance test, my father said that don't be disappointed. There is one more field where you can contribute, and that is in agriculture. So coming into agriculture was just because of my father. And after coming into agriculture, once I decided, I made up my mind, okay, this is also a good field where I can contribute. Then I gave my best to this field. And I always stopped in my studies and I used to do whatever best I could. And when we came for post-graduation, genetics and plant breeding was the most sought after discipline, because we see that not to belittle any strain or any discipline, but most of the successful things have come from plant breeding. And for plant breeding, you need to have a comprehensive knowledge of all the fields. Genetics and plant breeding, they go hand in hand together. So we don't take them as two different fields. We take genetics and plant breeding together into one. And that is how I started my journey for agriculture in 88. I joined my BS Agriculture. 92 I completed, then 94 I completed my Masters in Genetics. And 95 I joined for Ph.D. in Genetics at Indian Agricultural Research Institute. I got good training from my teachers. My foundation for genetics and plant breeding came from my graduation and post-graduation teachers. And then it was again polished under the guidance of able teachers at IARI. And the kind of exposure we got at IARI is uncomparable, because there the faculty, they used to do research, they teach us, and they train us in such a way that we learn and also we try to deliver it. So it is a very good kind of training we got in IARI. And all being in Delhi, many people used to come from worldwide, and we used to get good exposure, and we used to be, means, we used to imagine how we should be, then what we should be like. So that's how I came into this field, and then cleared in ERS, Agricultural Service. My first posting was in Soybean in Indore.
Okay.
I worked for about 7.5 years. After that, I joined again in IRS senior scientist. From then I was working in Pearl Millet. In 2017, I joined as project coordinator for AICRP Pearl Millet. After joining immediately, this benchmark thing has come, and we implemented this. And that is how we started taking lead in many things.
Wow. Nice. Ma'am, my next question is that according to you, which sectors of plant science research and agriculture will prosper in the near future?
Land bidding, no doubt, ma'am. this biotechnology thing. See, any discipline you work in, ultimately you have to get a technology or a product.
Right.
The kind of technology is product. That is your variety. You have very good varieties in rice, in wheat, in pearl millet, many things. Without the variety or the material, you cannot get yield.
Right.
With the help of different disciplines, we can develop, integrate all those information and come out with variety. So, in my opinion, breeding is the best, then followed by biotechnology.
Okay, ma'am. My next question is that how can we translate our knowledge of plant science into food security?
This is what we're doing. Today,
Yes.
my that only. Knowledge we gain, that is what we're trying to translate. We know about biofortification, we know about the advantages of these micronutrients, and that is why we try to bring that science and make it part of the breeding program and then bring it in food security. I said food security along with nutritional security.
Yes, yes, yes. Yes, ma'am. Ma'am, my last question is that what would be your word of advice be to the young women in their early PhDs who are aspiring to be plant scientists?
Yes, this is a very important question. And anywhere you go without hard work, you cannot come up. Young women, they should be prepared physically, mentally, psychologically, and emotionally. In all these aspects, they should be very strong. And in the present condition, the women are faced with many issues. I don't say men are not having any issues, but women are having many problems. See, they have, first of all, though we are talking of equality, gender equality, still we find a unseen kind of pressure on the girls. They are not allowed so freely to come out. Whether if they are allowed also, the women also, the girls also feel insecure because of the existing conditions in the society. So
Right.
to prepare ourselves to overcome all these unseen hurdles, then for women, young women, I say that economic independence and mobility, independent mobility is a very important. If you're economically independent and if you're really mobile, to go on your own the things, this will solve most of the problems. And anywhere you are, you have to do hard work. Without, there is no shortcut to hard work. And in this competing world with males, women have to run twice or thrice more compared to males to show that they are better.
So true.
I'm sorry to hurt any.
Thank you, ma'am. It was wonderful. Your presentation, the way you talk so passionately about your work is amazing. Thank you, ma'am. I'm giving it over to Shubho.
Thank you, ma'am, for your talk. Now, I'm just a request because today is our woman speaker, so I request Neha and Jacinta to say goodbye to ma'am. We hope in future with many more varieties and many more biofortified iron, zinc, and vitamin millet will come to the market, and we are happy to have this type of food because in future food crisis is a major concern, so we have to little bit change our food habit also, and we should consider these food as an alternative, and these are more nutritious and can be supplemented with our normal rice, wheat, maize. So I request Neha to say say goodbye to ma'am. I request to Jacinta, goodbye ma'am. Ma'am, we going to some our formalities like how to collect the certificate and that all. And with that note, you can leave the meeting. And
Thank you.
I think Neha and
Thank
Jacinta
you so
will.
much, ma'am, for your inspiration to the young scientists.
Yeah, and yes, thank you for inspiring us and telling us about your great research and progress into biofortifying these crops. I'm really excited to see how it all works out, and I hope that it's up, you know, it's taken up well in the communities and it makes a big impact for everyone in nutrition. So yeah, thanks again for joining us and being part of our platform. And I think, yeah, and answering all the questions in question time so well. Thank you.
Thank you so much.
Thank you, ma'am.
Thank you very much.
So now, Shoma, you going to share your screen and give some instruction to our audience.
Okay. Now that we have officially come to the end of our webinar, I will give some instructions, important ones. I'm sharing my slide. So I hope all of the participants, registered participants, have received the password, and I think many of them have hurried to the website to collect the certificates. The next webinar is on 13th July. It's on understanding plant-virus interaction to identify novel antiviral weapon in the plant arsenal by Dr. Vasudev Kaushal. Then again, we have another webinar on 15th July on plant viral diseases, their economic implications and their management by Dr. Vasava Prabhu L. Patil. Next again on 17th, on the topic, role of reactive oxygen species and antioxidant machinery in crop plants by Dr. Tahmina Islam. She'll be joining us from Germany. Then again on 18th July by Dr. Deepak Gayan. He'll be joining us from Rajasthan, India. He'll be talking on improvement of storage stability and productivity of rice through genetic engineering. The next, our webinar is on 21st July on climate smart rice hybrids by Dr. Jauhar Ali. He'll be joining us from Philippines. He'll be talking on 21st July. Okay, next. A new webinar has been added on our website. The speaker is Dr. Raviraj Banakar. He will be joining us on 24th July. The registrations are just open just now. It's a new one. He has been in the University of Minnesota, USA, and he recently joined Avastagen Limited, India. Please register for this. It's a new registration. And once again, I want to say that only the registered candidates will be entitled to receive the certificate of attendance. Regarding the certificates, we have devised a new certificate distribution system. In the previous one, what we were doing, we were mailing the certificates to each applicant, but it was taking a lot of time because there were more than 1,800 or 2,000 applicants there. So what we did, we devised a new one. Now we are creating all the PDFs of the certificates, and we are uploading it. And we are sharing it. We are sharing the drive link to you on our website. Go to our website. Okay, I have the screenshot. Go to our website. On our website, you will find that there's a certificate collection link. Okay, there you can collect the, you can fill out the certificate application form and collect the certificates. And after that, After that, you are going to get a link on the website. That link will consist of the list of all the applicable or the applications that we found, and you can search your name from there. It's in alphabetical order. You can search your name from there, and you can find that there is a file number assigned to your name. Then go again to our website and you can find that there's a certificate collection link. Click on that and you will find there are all the file numbers are given in the PDF format. Give the page to some time to load. The page will load and then you will see that all the file numbers are there starting from 1. Please find out your file number, download the file. There you have your certificate. So no more waiting. You just have to wait for 2 days after each webinar so that we can do this, upload the files on the website, on the drive. Okay. See you at the next live webinar on 13th July at 11 a.m. Please register from our website. Thank you.
Thank you. Thank you for the discussion. Thank you, madam. And thank you, Neha. Thank you, Jessica, for joining us. It's a nice pleasure to all of you, and we successfully completed one another. We will meet in 13. 13 for our next webinar, and I wanted to just add one thing. There's now onwards, because there is lot of certificate and lots of participants are interested now. So we now we decided that only those are pre-registered and also attend the webinar will get the certificate. But our webinar is a free for all. If everybody can see it and learn something from the webinar, but it's for the certificate, he or she must have a registered participant, and we.
Then,
Yes.
Yeah, you finish the analysis.
Okay. So our next webinar has we have a 3 to 4 days, and our registration is ongoing. So I request every listener or viewers that go to our website and register what webinar you want to register, because there is a lot of webinar, and throughout the year there will be many more. So for registration, they should register yourself first, and when we next webinar, when we getting the application, the application, after the application we tally our registered webinar list and the applicant list, and those are the matched will get the certificate. So we open the feedback option to everyone, but for the certificate you have to pre-register. All registration is available, and one registration start today, as Shoma says, that because the speaker is in a transition period, so now he is able to deliver the webinar. So it is on the CRISPR. CRISPR-Cas editing, most of the talk based on the plant, but little bit other system cell type will be added. So so this webinar registration is just begun, and we already got about 1100 application for today's webinar certificate. So we are uploading a list and the certificate too as soon as possible, and I hope all applicant will get the certificate. And I repeat that every certificate is on the drive, so don't be panicked. It is sometimes it's not visible due to low internet speed, but if you have an internet, good connection of internet, you can see it. Because when I upload it, I checked myself, I checked whether all the name of the participants and number of certificate is there. Then only I shared the drive. So there is no chance of that their name is not there. Yes, if you unable to apply the form, there will be a chance that you can't get your name on the list and the certificate. And this link active for 2 hours up, and I today at 2:30 p.m. in Indian Standard Time, it will disappear from the everywhere. So my request to everyone, go register yourself for next upcoming webinars, apply for today's certificate, and we'll meet again in 13 for the next webinar. It is from Vasudev, Dr. Vasudev, University of California, USA. So I officially I just finished this meeting. Thanks, Jacinta. And I think Neha is not there. So I officially end this meeting. Thank you. Okay. তোমরা চলে যাচ্ছ তাই তো? Here from here, the certificate will be available. This is the section in bottom of the home page of our website. These are the last webinar certificate, and very soon there is another link with the drive link for today's webinar certificate. This particular webinar just open for registration. Go and register for it. These are the upcoming webinars. We just completed this one, and next is the webinar on the plant virus interactions. We got the application about 1,185 till this form is active up to 2:30 p.m. Indian Standard Time. We collect the name of the applicant and make the certificate and upload to our website here. Thank you, everybody. I'm officially end this session, close the Zoom, and YouTube video. I'm now make it private, and very soon, within 2 days or 3 days, I will upload all the webinar videos, interview videos to our YouTube channel. I request all of you, please like and subscribe our channel so we have no problem for giving the intimation or the notification. You automatically got the notification when we start a live session. So it's make our job easy to inform everyone, because our webinar is free to everybody, open to all. But as I said, from next webinar, only registered participate and attendees getting the certificate. Your registration your registration is not your certificate. Certificate application is a separate, because there is so many people. Those are not required such certificate, but listen the webinar to increase their knowledge. So I request everybody go and register upcoming webinars so we can understand that how much attendees are excited, because it's also help to the speaker to design his or her talk, because we getting the information of your profession, that how much BSc, how much MSc, how much PhDs, how much scholars or professionals are going to attend this webinar, and our talk designed in such a way. So registration is required for me, and also registrations are also required to understand you more, because this form is more elaborate. Also, we need your feedback. So we going to post as like we posted the link and password in all upcoming webinars, but only pre-registered and attended candidate will get the certificate. We have time. There is a 3 days gap, so go and register. In 3 days, I will complete all the certificate distribution of first 2 webinars, webinar 1 and webinar 2. I think by tomorrow, we will complete our first round. Then we check whether the mail is bounced or not, whether you getting the certificate or not. If you have any question, mail us. Don't WhatsApp us, don't write on the Facebook Messenger. Officially and formally mail us. When you mail, please mention the exact webinar name and date, because we going to organize so many webinars and we sometimes missed with another. Your name should be on the list. Otherwise, your message or your request will not entertain. Your name should be on the list. If you're not getting the certificate, we can do whatever we able to do, but you have to the name available very soon after fusion. If you're not getting it, check your internet, check your Google account, Drive login, but your certificate is there. Don't contact us for this purpose. If your name is not in the list, we assume that you are unable to apply. This type of message, we require your feedback, copy of your feedback form, which was given by the Google automatic service. So we need that form to confirm that whether you apply or not. And don't give so many excuses. I know I know the excuses. So your internet speed, your system, your problem is not going to entertain. You have to attend, you have to apply, your name should be in the applicant list, you get your certificate. That is. Okay. See you all in next webinar on 13 at 11 a.m. Indian Standard Time.